In-vitro release testing, or IVRT, is a crucial step in pharmaceutical development. It helps us understand how a drug is released from its dosage form. This information is vital for ensuring the drug's effectiveness and safety. Essentially, IVRT mimics what happens to a drug inside the human body after it's taken. Think of it like this. When you swallow a pill, it doesn't just magically release the medicine all at once. The pill's outer layer dissolves, and the drug inside gradually enters your bloodstream. IVRT helps us recreate and study this process in a controlled laboratory setting. By analyzing how a drug is released, we can predict how well it will be absorbed by the body. This helps us determine the optimal dose and dosage form for a particular drug. IVRT also helps us identify any potential problems with the drug's formulation, such as poor dissolution or instability. Overall, IVRT plays a critical role in ensuring that new drugs are both effective and safe for patients. It provides valuable insights into the drug's behavior, allowing us to optimize its delivery and maximize its therapeutic benefits. Choosing the correct receptor media is crucial for successful IVRT. The receptor media is the fluid that mimics the environment the drug encounters in the body. This could be the fluids in the stomach, intestines, or even the bloodstream. The right receptor media depends on the type of drug and its intended route of administration. For example, if we're testing a drug designed to be absorbed in the stomach, we would use a receptor media with a pH similar to that of gastric fluids. Alternatively, a pH 7.4 phosphate buffer, ethanolic solutions, or organic solvents could be used based on the dissolution of the drug in the receptor fluid. For topical drug products, the receptor fluid for IVRT studies should closely mimic the conditions of the skin or mucosal surfaces. Selecting the appropriate receptor media ensures that the IVRT accurately reflects the drug's behavior in vivo. This is essential for obtaining reliable and meaningful results that can guide drug development and ensure patient safety. Just like choosing the right receptor media, selecting the appropriate membrane is crucial for successful IVRT. The membrane acts as a barrier between the drug and the receptor media, simulating the biological membrane the drug needs to cross to be absorbed. Different membranes are available, each with unique properties that mimic different biological barriers. For instance, synthetic membranes like cellulose acetate are commonly used to simulate the intestinal lining. In contrast, natural membranes like pigskin might be used for topical or transdermal drug delivery studies. The choice of membrane depends on several factors, including the drug's properties, its route of administration, and the specific research question. Selecting the correct membrane ensures that the IVRT accurately reflects the drug's behavior in vivo. This, in turn, leads to reliable and meaningful results that can guide drug development. Franz diffusion cells are essential tools for conducting IVRT. These specialized pieces of equipment provide a controlled environment for studying drug release from a dosage form. Imagine them as tiny chambers with two compartments separated by the membrane we discussed earlier. The dosage form, such as a tablet or cream, is placed in the donor compartment. The receptor media, simulating bodily fluids, is placed in the other compartment. The membrane separating the two compartments acts as the barrier the drug must cross. Over time, the drug is released from the dosage form and diffuses across the membrane into the receptor media. We can then analyze samples taken from the receptor compartment at specific time points to determine the drug's release rate. Franz diffusion cells come in various sizes and configurations, allowing researchers to tailor the setup to their specific needs. They provide a controlled and reproducible environment for studying drug release, making them indispensable tools in pharmaceutical development. Choosing the right time points and speed for an IVRT study is crucial for obtaining meaningful data. The time points determine when we collect samples from the receptor compartment of the Franz diffusion cell. These samples are then analyzed to determine the amount of drug released over time. The frequency of sampling depends on the drug's release profile. For example, a fast-releasing drug might require more frequent sampling in the initial hours of the study. In contrast, a sustained-release formulation might only need sampling once or twice a day. The speed of the experiment, specifically the stirring speed in the receptor compartment, also plays a vital role. Adequate stirring ensures uniform drug distribution in the receptor media, preventing concentration gradients that could affect the result's accuracy. Determining the appropriate time points and speed ensures that we capture the complete drug release profile. This information is crucial for understanding the drug's behavior and optimizing its formulation for maximum therapeutic benefit. Mounting a membrane on a Franz diffusion cell is a delicate but crucial step in IVRT. Proper membrane mounting ensures that the barrier between the donor and receptor compartments is uniform and without leaks. This is essential for obtaining accurate and reliable results. The process begins by carefully placing the chosen membrane between the two halves of the Franz cell. It's vital to avoid any wrinkles or air bubbles that could create channels for the gel or ointment to leak through, bypassing the membrane. Once the membrane is in place, the two halves of the cell are securely clamped together, ensuring a tight seal. This prevents leakage and maintains a constant diffusion area throughout the experiment. After assembling the cell, it's essential to perform a leak test. This involves filling the receptor compartment with a suitable solution and visually inspecting for any leaks. A properly mounted membrane should prevent any fluid from passing through, guaranteeing the experiment's integrity. Before using an IVRT method for routine testing, it must be validated to ensure it is accurate, reliable, and reproducible. Method validation involves performing a series of tests to demonstrate that the method consistently produces results within a predefined acceptable range. This typically involves analyzing samples of a known concentration of the drug and assessing the method's accuracy, precision, linearity, range, and robustness. Accuracy refers to how close the measured values are to the true value. Precision measures how close repeated measurements are to each other. Linearity assesses the method's ability to produce results proportional to the drug concentration over a specified range. Range defines the upper and lower limits of drug concentration where the method performs accurately and precisely. Robustness evaluates the method's susceptibility to minor variations in experimental conditions. A validated IVRT method provides confidence in the results obtained, ensuring they are reliable and can be used to make informed decisions about drug development and quality control. Bioequivalence testing is crucial for demonstrating that a generic drug product releases its active ingredient into the bloodstream at the same rate and extent as the brand name drug. This ensures that the generic version is therapeutically equivalent to the original and will have the same clinical effect. IVRT plays a vital role in bioequivalence testing by comparing the dissolution profiles of the test, or generic, and reference or brand name drug products. The goal is to demonstrate that the two products exhibit similar drug release characteristics, indicating comparable in vivo performance. Regulatory agencies such as the FDA set strict criteria for bioequivalence, requiring the generic drug's dissolution profile to fall within a specific range of the brand name drug's profile. This ensures that patients switching to generic versions can be confident they are receiving a therapeutically equivalent medication. Once an IVRT study is complete, the data collected needs to be analyzed and interpreted to understand the drug release characteristics. This involves plotting the amount of drug released over time, creating a dissolution profile. The dissolution profile provides valuable information about the drug's release rate, allowing us to determine if it's immediate, delayed, or sustained. This information helps assess the drug's suitability for its intended use and optimize its formulation. In bioequivalence studies, the dissolution profiles of the test and reference products are compared statistically. This involves calculating parameters such as the area under the curve, or AUC, and the time to reach a specific drug concentration, or Tmax. These parameters are then compared using statistical tests to determine if the two products are bioequivalent. If the test product's dissolution profile falls within a predefined acceptable range of the reference product's profile, the two products are deemed bioequivalent. Confidence intervals, typically 90%, are calculated for the difference in key parameters between the test and reference products. If the confidence interval falls entirely within the bioequivalence acceptance limits, it provides strong evidence that the two products are bioequivalent.